Good morning to one and all. Thank you so much for joining us. And so today's speaker is Dr. Pradeep Kumar Singour. So I just want to give a brief introduction about Dr. Pradeep Kumar Singour, sir. So, Dr. Pradeep Kumar Singhal is a highly accomplished pharmaceutical scientist with an extensive academic and research career spanning over 20 years. Currently serving as a professor and vice principal at Venus Group of Institutions, Faculty of Pharmacy in Bhopal, Madhya Pradesh, India. Dr. Singhal is renowned for his expertise in pharmaceutical chemistry and drug design. He completed his B Pharm and M Pharm in pharmaceutical chemistry from Dr. H.S. Gaur, Central University, Sagar, and later earned his PhD in pharmaceutical sciences from the same institution. Throughout his career, Dr. Singour has made significant contributions to the field of pharmaceutical research with a particular focus on rational drug design. His research interests encompass various areas including virtual screening, de novo design, QSAR, analysis, molecular docking, conformational analysis, biochemistry, and medicinal chemistry. He has authored over 43 research and review articles published in prestigious national and international journals, demonstrating his commitment to advancing pharmaceutical science through cutting edge research. Dr. Singh's dedication to teaching and mentoring is evident through his supervision of more than 40 MPharm students, the guidance of two PhD students in their research projects. He has recognized for his outstanding contribution to education and research, receiving awards such as the Various Employee of the Year Award in 2019, the iconic Faculty Award from Shiksha Sansthan Jaipur in 2019, and the Excellent Teacher Award in Pharmacy by Sister Nivedita Takniki Shiksha Samiti Bhopal in both 2013 as well as in 2018. Additionally, Dr. Singour has shared his expertise by delivering expert talks at various conferences, serves as reviewer for reputed international journals, further establishing his prominence in the field of pharmaceutical sciences. We all welcome you, sir, for the today's webinar on computational approaches streamlining drug discovery. So, welcome you, sir, and thank you so much for joining and giving your precious time and sharing your time with us. Thank you so much, sir.
Uh, Thank you, Mr. Monu, and thank you for introducing me among the participants. Uh, Today, we will discuss about the different computational approaches that will streamline the drug discovery, because we know what are the drug discovery, and there are different steps involved in drug discovery, where the timeline of the discovery is high. So, by the help of different computational approaches, can we cut the time, and also we can reduce the time, and as well as reduce the cost. So here we will discuss only some brief of that computational approach, because there is a wide areas of the data discovery and the computational approaches. But here we will only that uh, discuss some approaches. What are the approaches, and I will give some uh, how we can perform the computational approaches. So we will discuss, uh, as we know that the life expectancy is now increasing, because there are different drugs have been discovered, and also there are the new drugs have been discovering. So we have to involve in different approaches of the computational approaches, because the era is the era of the computational technology. So we can combine the computational as well as the technology, so we can discover more new drugs, so we can all increase the life expectancy also. So, and we know, I think all we know about the what are the adventures in drug discovery. For the development of the one drug, we have to start from the 10,000 to 30,000 compounds, and we have to synthesize, examine, and then screen, and then only 10 to 20 compounds come into the preclinical tests, and then we have performed the different clinical trials on phase 1, phase 2, phase 3. and finally we got only one compound. So the time required for the discovery also approximately 15 years, and the cost of the compounds will for the production, for the development, will also high, because we have to start from the 10,000 30,000 compounds, and only we get only one successful compounds. So how we can cut the time and how we can reduce the cost by help of that computational approach, we will discuss today. um, I already discussed that any drug come into the market after starting from thoughts and starting from 10 to 12-year back, 15-year backs, and we reached only the one compound, that approved compound. So, what are the different challenges of the medicinal chemist? Because that if you want to discover any one compound, that they have to increase the costs and enormous time. That we can, by the help of computational approaches, we can, how we can develop the successful compound by reducing the time. So these the drug discovery and development time, how we can discover that. First we have to identify the disease, and then these are the traditional approaches by which, first we have to identify the disease, and then we have to isolate the proteins that involve in particular disease that require 2 to 5 years, and then we have to find the drug that is effective in that particular disease and particular proteins and particular receptors that require 2 to 5 years, and then we have to perform the preclinical testing that's also required some years, and then we have to perform the scale up, then file IND, and then we have to perform the human clinical trials, and then file NDA, and then require the FDA approval of the drug that require 2 to 3 years. So we can see here there are the lots of years are required to develop any drug discovery. So how we can reduce some years by the help of that some molecular modeling techniques and the virtual screening, that's in to find the different drugs that is effective in the particular disease. So what are the different molecular modeling techniques and what are the different virtual screening techniques? Molecular modeling, by name itself, that's modeling of the molecule. We know that modeling, we it is a general words, but more, if you perform the modeling of the molecules, it means the molecular modeling. And by the help of virtual, by the help of computer, if we screen the lots of compound, that's known as a virtual screening. So by the help of virtual screening and the molecular modeling techniques, we can design the drug, and we can get the effective drugs in very lesser time. So what are the different computational approaches? Because we are discussing today about the computational approaches that streamline the drug discovery, means how we can drug discover by the help of that computational approaches. So where we can involve, so we can see that if you want to isolate the proteins, so by the help of genomics, proteomics, biopharmaceuticals, we can um, isolate the proteins by the help of that study of the genes, that genomics, study of the protein, that the proteomics. So by the help of these methods, and also by to find the drug, so we can screen the 1 lakh compounds at a time. That is high throughput screening. That lot of compound we can screen which compound will be effective and which compound is not effective. So we screen by the help of high throughput screening, and virtual screening by the help of computer. Virtually we can screen the lot of compounds, and how we can reduce the time of that synthesis by the help of computational chemistry. So computational chemistry, computational chemistry. So computational chemistry, it means that if we synthesize the compound in a combination, so we can reduce the steps. Suppose we want to synthesize the peptides. So peptides require that combination of the different amino acids. So can we reduce the time of that coupling of the reactions, of the removal of the coupling agent, removal of the protecting agents? So we can combine these steps in a one steps, so we can reduce the time. So that is known as a combinatorial chemistry. It means that chemistry we can perform in a combination. So we can reduce the time. It will help to reduce the cost of the development of the drug. And that molecular modeling, we already discussed that modeling of the molecules by the help of computers. So we can develop drugs. And also, there are different models in silico model of the ADME. We know the ADME, that's absorption, distribution, metabolism, and excretion. So there are different model like CCADME model, and then also there are some toxicity models are also available in which we can determine their absorption, pKa value, also that metabolism, also that their toxicity profile. So all we can there are different softwares are available, so we can screen out the compound which will absorb in the body or not. So now we come into the different drug discovery process. First, we have to we that discussed earlier that first we have to identify the targets, and then after identification we have to validate the target. Then we can perform the high throughput screening. Then after throughput screening, we can um, discover the lead compound, and then after evaluation of the lead compound, and then we validate the lead compound, and then finally we can come into the clinical trials. So after completion of the clinical trials, we can get the most effective compounds. So these are the process. And also the high throughput screening can be covered by the chemical library. We can use the chemical library that by the natural products. So natural product chemical library of the compounds that can be help into the high throughput screening. And drug discovery process, we discussed that by the chemical library help of the chemical library, we can use the rational designing, and then molecular modeling approaches, and then virtual screening, and the early ADME and toxicity profiling. So these are the different computational approaches by which we can develop a new drug. So where we can use and which type of techniques. So suppose there is a therapeutic target is there. The therapeutic target, how we can identify the therapeutic targets. So by help of that functional genomics, and then structural genomics, and then proteomics. And for the functional genomics and structural genomics and the proteomics, there are different softwares are also available by which we can develop therapeutic targets. And suppose you want to develop that, you identify the targets, and then you find the novel ligands, and how we can find the ligands, then by the help of structure based drug designing, and then chemoinformatics and high throughput screening. And and then suppose you come into the clinical candidates. So clinical candidates, you have to perform the in vivo disease model. There are different type of models are available. And then admit and drug delivery. Admit means ADME, that is absorption, distribution, metabolism, excision, and T means toxicity. So these models are also available. And then we can come into the medicine, that find you the best medicine. So these are the different approaches, and each approaches are used for the different purpose. Someone is used for the uh, development or identification of the target, someone is used for the ligands identification, someone is models are used in the clinical trials. During the clinical trials are the clinical candidates, that's in vivo disease model, admit, preclinical. So there are different approaches. Here I am only giving some brief, actually, just these are the methods, because every methods have their details, and we cannot discuss here because and there are different steps are involved for the every approaches. So only we are here discussing about the approaches only. And molecular modeling approaches. So, molecular modeling means the modeling of the molecule, and by the help of different approaches, just like that NMR and X-ray determination. By the help of NMR, X-ray structural determination, we can identify the proteins, because first we have to isolate the proteins, and then we have to determine the structure of the proteins. So if you want to determine the structure of the protein, we can determine by the help of NMR techniques and X-ray determination. And then we have to suppose we have to perform the energy minimization of that particular proteins of the or the ligand. So we have to perform the QM/MM method. QM means quantum mechanics, and MM, that is a molecular mechanics method. So quantum mechanics and molecular mechanics method, these methods are used to generate the different uh, candidates or the ligand, that by the help of energy, we can say that we can reduce the energy, we can minimize the energy by the help of these methods. So we can get the low energy molecules. Why we require a low energy molecule? Because it is a stable molecule. Because when drug is interact to the receptors, it should be in a low energy state. So how we can perform the energy minimization by using this energy minimization calculation, energy calculation method, that is quantum mechanics and molecular mechanics. And there is some homology modeling. Right side, we can see that homology modeling. Suppose you don't know the actual structure, yeah, how we cannot determine the structure of the protein by the help of NMR X-ray, then we can perform the homology modeling. If you know the structure of the receptors of the same family, so we can design the homologous structure of that same family. So we can draw manually the structure of the receptor by the help of homology modeling. Then we can perform the further study. And here we can say in upside that QSR and 3D QSR, that is ligand based drug design process. That QSR, that is quantitative structure activity relationship. If we have the large number of compound in the same series, so we can perform the SAR, but quantitative SAR. So we can get one equation, and by the help of these equation, we can determine the biological activity of the unknown compound. So later we will discuss in detail. And there is in low side, that is combinatorial chemistry. So already we discussed, if we perform the some reactions in a combination, so we can reduce the steps, that is known as combinatorial chemistry. And that is another one is bioinformatics and informatics, in which that is large number of molecules we can get their data by the help of bioinformatics. So what is molecular modeling? Molecular modeling, that is a visual representation of the molecule and their property, and by the help of these property, we can perform the drug designing process. And computational approaches and chemistry, that is simulation of that molecular property of the compound through the different equations. These are the equations, and we have to perform the statistical modeling also, and then there are different informational management of the database of that large number of the property. So these are approaches also used in when we perform the molecular modeling. Okay, so which approaches will use when? So we can see this table, in which so when we see that drug receptor interactions, there is some receptor is there, and some is drug or a ligand is there, and the drug or ligand is bind to the receptors. So suppose there is you don't know the structure of that receptor, and you don't know the structure of the ligand, then which method you follow? So we can see in the first column is there. So we have to perform that high throughput screening and the combinatorial chemistry, in which we can synthesize a novel compound in earlier step, and we can screen the large number of compound, which compound will act to the will receptors. So this method is used when you don't know the structure of the receptor as well as the structure of the ligand. And right side, you can see that if you know the structure of the receptor, but you don't know the structure of that ligand, then you can perform the de novo drug designing. Later, we will discuss in detail de novo drug designing, in which that if you know the structure of the receptor and we uh, build the structure of the ligand inside the receptor, that is known as de novo drug designing. So later we will discuss in detail. In this, we can see that suppose you don't know the structure of the receptor but know the ligand structure, so we can perform the ligand based designing. Ligand based designing, that QSR approach, pharmacophore based approach. So QSR, also 2D QSR, 3D QSR. So these approaches can be used. These approaches develop by equation, and by the help of equation we can later, by the help of equation we can uh, estimate or determine the biological activity of the unknown compound. And here the last one, if you know the structure of that receptor as well as the structure of the ligand, so we can perform the molecular docking. Docking means you can, in general word, fitting. That there is one structure of the receptor and there is one drug, and drug will fit to the receptor. So by which we can see that what are the different bonds are involved to the fitting, and how much that fitting is there. It is good fit or not fit. So that is we can see from the molecular docking. So we can screen the compounds by the help of molecular docking techniques also. Here also some more techniques which or we can see computational approaches that's used in a different purpose. Suppose that if you want to calculate the physiochemical property of that compound, so we can, by the help of there is some software and available, its apps are also available, in which you can calculate the log p, dissociation constant. And for the drug designing, there are ligand based drug designing. The ligand based drug designing is performed by the help of QSAR, pharmacophore based drug designing. And structure based drug designing, that is by the help of docking, de novo drug designing. And there is some pharmacokinetic modeling also, by which we can determine the absorption, metabolism, distribution, toxicity profiling of the drug. And chemoinformatics, by chemoinformatics, that is database management, similarity search, different methods are available for the chemoinformatics. So all other, if you join all the methods, so we can perform the virtual screening protocols, so we can develop new drugs. Here I am giving some name of the softwares that is used for drawing tools by the CameDraw, Marvin Sketch, CameSketch. And these some softwares are freely available in the internet, so you can just download and then we can use by the help of CameDraw, Marvin Sketch, CameSketch. So these CameSketch, these so we can draw the structure, because if you want to molecular modeling technique, if you want to perform the QSR approach, you want to perform the docking approach, then first we have to draw the structure. So by the help of these software, we can draw the structures. So you can just write down all the name of the softwares by the help of which we can draw the structure of the compounds. These are the some molecular modeling techniques or software, that is charm, that is bromex, amber, cis parameter, then cis side chain. So there are different techniques or software, you can say, that by the help of these we can perform the molecular modeling. For the binding site prediction, uh, suppose you know the structure of that receptor, and you find out the you want to find out the binding sites. So by the help of these different softwares, you can predict the binding site. So you can just write down docking. So I'm just discuss what is docking. So for the performing of the docking, we can use Autodock software, Dock, Gold software is there, and docking server. Schrodinger software is also available in which we can perform the docking study. So kind of target prediction, and these are the softwares. ADME toxicity, we can perform by the Volsurf, GastroPlus, MatCam Studio, LOPS, CCADME. CCADME is freely available. You just write down the CCADME. Just you can type the CCADME in the Google, and just you can see the one website is there, and you put the structure, and then you uh, these are the different softwares. Now come into that QSR. Here I am discussing few techniques in detail. So QSR, that is quantitative structure relationship, in which we have to develop a mathematical relation. We have to develop an equation between the biological activity and the physiochemical properties. So we have to generate the equation by the help of physiochemical property. There are different physiochemical property like the lipophilic property, steric property, hydrophobic property, electronic property. So these first we have to calculate the these property of the compound, and then we have to develop the equation in between the biological activity and the physiochemical properties. So there are different type of QSR, that is classical Hens analysis, 2D QSR, 3D QSR, and 3D QSR, molecular field analysis, molecular shape analysis. So there are different techniques are available. So in QSR, suppose you want to perform the QSR study. So these are the steps of the QSR study. First, you have to select a training set. First, we have to identify a series in which there is a more than 20-25 compounds should be there, and we have to divide the data into two set, test set and training set. And first, so suppose you select the training set, then you enter the biological data in the software, and then we have to perform the energy minimization property. Energy minimization, that is generate a conformation, that is energy minimization, energy minimize conformer. And then we have to calculate the descriptor of all the energy minimized molecules, and then by the selection of statistical method, we have to generate QSR equation. And then after generation of the QSR equation, then we have to validate the QSR equation by the help of test set. We can generate the model or equation by the help of training set, and then we can validate the QSR equation by the help of test set. And then finally, uh, if you develop the QSR equation and validate the QSR equation, then you can predict the activity of that unknown compound. Unknown compound, we can predict the help of that QSR equation. So here I'm just showing how we can draw the structure, and so we can draw the structure, we can draw the benzene and then bonds, and we can draw the structure. Then finally we can come into that came 3D software, and here we can minimize the energy of that molecule, and then we can find after then we calculate the properties. Here is some list of that properties, and then alpha polarizability, band energy, boiling point. There are different properties, so we can calculate, and after calculation we can develop the QSR equation. So what are the different properties? What are the different descriptors there? So there are different descriptor like structure descriptor, electronic descriptor, thermodynamic descriptor. So there are different descriptors are available. Descriptors means properties. So we can calculate the different properties by the help of particular software, and then we can develop this type of QSR equation. So in which log 1 by C, it means that biological activity, and we can develop a QSR equation and biological activity and physiochemical property. So by the help of those, so we can see here some physical property will have the positive correlation, some will have the negative correlation. So these are the this is the QSR equation. So in which we can uh, calculate the biological activity means log 1 by C of that unknown compounds. If you know the pi value and the sigma value of that particular unknown compound, then we can put the value in pi and sigma, and then we can calculate the biological activity. Molecular, that is field analysis, shape analysis, that is molecular field analysis, in which that we have to align all the molecule, energy minimize molecule, and then we have to all the put the all molecule in the grid box, and then we can calculate the field, and that is known as a comparative molecular field analysis. In short, that is COMFA analysis, that is a CDQSR approach, in which we can calculate which area of that molecule is prominent. Suppose that is red color is there, so we can see here the red is indicated electronegativity. It means there is a, if you increase the electronegativity at particular area, so we can develop a potent compound that have the maximum activity. In blue color indicated that is lactopositive, it means you can put the you can add the group that have nature of the lactopositive. So if you add in this area, so we can develop a novel compound. And just like we calculate the steric field also, in which green color indicated someone, and that yellow color is, it is the repulsive steric field. So by the help of this color, that is known as a contour map, in which there is contour color is indicated. Yeah, we can say there's contour maps by known as contour maps. So by the help of that contour maps, we can uh, add particular group at particular area, so we can develop a best rep. So first we have to develop these as a contour maps by that help of that comparative molecular field analysis. And some more we can see here the some more properties there, steric property and the electrostatic. First we have to align, and then we can calculate the field. And also that COMSIA analysis also now that is comparative molecular similarity index analysis, in which also use that properties, these properties also. So another technique, that is a pharmacophore approach. And pharmacophore approach, there are different type of pharmacophore approach. Pharmacophore, it means the active part of the molecule that is responsible for the pharmacological activity. So by the pharmacophore approach, we can identify the active parts, identify the pharmacophore parts, so we can develop a better drug by the help of these pharmacophore approaches. So these are the different steps of that pharmacophore process. We can by the training, first we have to training set selection. Just I have discussed in a series, we can divide the test set and training set. Then after training set selection, then feature selection, which features you want, and then conformation generation means energy minimization generation, and then alignment of the all the molecule. Alignment means you have 20 compounds in a training set, so you have minimized the energy of the all the 20 compounds, and then we have to align all the molecule over to each other. Then after alignment, and then we can develop a model, pharmacophore model, and then by the help of that pharmacophore model, we can develop a new drug. So this is known as a pharmacophore approach. And pharmacophore features, there are different uh, uh, HB receptors, donor, hydrophobicity, hydrophobia, aliphatic, aromatic. We can judge, check, we can calculate that positive, here is a positive charge, negative charge. So by the help of these features, we can develop a model, and we can calculate the develop new compounds. So these are the steps of the pharmacophore generation. First, we have to input the structure, then conformation modeling, and then we can, with the SAR data, we can generate the model, and finally, we can evaluate the hypothesis generated. Now, coming to the next technique, that is the docking techniques. Docking, it means fitting, we can say. So, first we have to download the proteins from, that is one website, that is RCSB, that is the protein data bank. Just you can type the protein data bank in Google, and you can download the structure of the protein, and then we can perform the docking study. Then when you download the protein, then we have to optimize the protein by removal of the water molecule, adding of the hydrogens, and then, so it means that is known as a preparation of the proteins. So first we have to perform the preparation of the proteins, and then we have to perform the preparation of the ligand, means drawing of the structure of the ligand, and then preparation of the ligands, and then we can perform the docking study. Docking study, it means that drug will interact to the particular binding sites of that proteins. You all know that protein is made from the amino acids, so that ligand will bind to the particular uh, binding sites, mean particular amino acids, and if you form the more hydrogen bond with that particular protein, it means drug is most active compound is there. So by the help of these docking study, we can determine which compound is docking well, or which compound is not docking well. So there is some docking score is generated in a docking study. So after that, just like that, you say the cricket score is there. Cricket score, more score is means more run is there. Just like that in docking is there. There is some docking score. So better docking score indicated that the compound is fitting good to the ligand, or fitting to the good fitting to the receptors. So by the help of that score, we can check which compound is most fitted and which compound is not fitted. Another technique, that is a de novo drug design technique. De novo drug design, suppose you know the structure of that protein or a receptor, but you don't know the compound, which do not know the structure of that compound. So we can build the structure of that ligand inside the protein by the help of de novo drug design. De novo design, suppose there is a structure is there. These are the, suppose this is a structure. So we can develop inside the structure of that ligand. First, we can add a small group inside the protein. If he is binding to the receptor, then we can add the more groups or a ring, and then finally we can get a final compound that is most fitted to the receptor. So it means that building up that ligand inside the pocket of that particular receptor is known as de novo drug design. So there are different softwares are also available. So these techniques are the computational approaches. By this, we can develop a new better compound. So and now, what is a virtual screening? So virtual screening, it means there is, suppose you have a large number of compound, and there is a large number of compound in a library. So if you want to screen out which compound is active and which compound is not active, So we can screen the compound virtually by the help of in silico models, or by the help of computer, you can say. So we can screen, that is known as virtual screening. So virtual screening, suppose you want to, there is some natural compounds is there, lots of compound is there, natural compound. So we can screen the compounds. Now, the virtual screening is also now helpful in the repurposing also, because you know the compounds already developed in the market, but it is used for the some other purpose. So we can screen that particular drugs for the another purpose also. So it may be act to the another receptor or a proteins also, and you can, the particular drug will can also be used for the another purpose. So by the help of that repurposing, we can reduce the time of that production, reduce the time of that synthesis of that particular compound, because that particular drug is already in the market, but we are developing that particular drug or drug for the another purpose, another disease. So these repurposing is also using this these days. So they are scientists are developing that, scientists are searching which already developed drug is using it for another purpose or not. So another technique, that is a pharmacokinetic modeling. Pharmacokinetic, we know well that ADME is there. So that property relationship, there is some structure property relationship or not. So here we can say that most of the drug in the market are withdrawn because there is some commercial reason, other case some shows the adverse effect, someone is shows that toxicity, but most of the drug is not in the market due to their pharmacokinetic property, or that means there is some problems in the pharmacokinetic property. Drug is most effective to the particular receptor or enzyme, but there is some lacking to the property of that pharmacokinetic. There is some problem in the pharmacokinetic property, which means ki, suppose drug is not absorbing inside the body, so it is not in the market. But if you check that particular drug is binding to the receptor or not, it is better drug, but it is not in the market due to their pharmacokinetic. So we can check that pharmacokinetic modeling technique, that through the molecular modeling technique, we can check the pharmacokinetic property. So we can work on that particular area. Either we can enhance their pharmacokinetic property by any way. So the drug can come into the market. So uh, you can see that, suppose there is compound is good. So you can see that is good ligand because activity is optimized. We have performed the docking study and we find that particular drug is most active, good active. So this is known as a good ligand. But when the good ligand is converted into the good drug, when the activity as well as the property both are optimized. It means that physiochemical property, pharmacokinetic property. So when we can say the drug is good, when the drug is binding to the receptor as well as it have a good pharmacokinetic property. It is good absorb from that inside the body. It is not eliminated the body, and it is that is bioavailability is good. So these are the properties also matter when we develop the drug. So we can, by the help of the different pharmacokinetic model, we can develop a good drug. So what are the different property profiling assay we have to perform? That is, we have to perform the solubility study, permeability, bioassay, blood barrier crossing or not, cytotoxicity, transporter, metabolite study. So these study we have profiling of the drug, because drugs should must be survive, that there are different in vivo barrier inside the body. So because you know that in a stomach there is pH is 2, so its drugs should be stable in stomach. Drugs should stable in the intestine. It should cross to the intestine, and it should circulate in the blood, and blood have the proteins, so it should we have to check that protein binding of that particular drug, and there are renal clearance is there, how much drug, how much time the drug available in the blood. So we have to check by the help of different techniques and the models in for the development of the better drug. So each state of the biology has a property issue, just discuss, because suppose you perform the high throughput screening to purity, solubility, identity. These are the issue when you are performing the enzymatic assay, that stability in the bioassay and solubility. And then as you perform the cellular assay, then permeability, solubility, stability. And suppose when you perform the animal study, and then the drug is you want to give to the human, to then we have to check that that plasma stability, J stability, solubility, permeability. So these are the different uh, method or a technique that use that particular assay, and then we have to check if particular drug is stable or not. So there are different models and approaches all can help to check these properties. So there are some successful drugs that came into the market after drug designing process by the help of different computational approaches. These are the drugs that discover for the different purpose. Just like that, erythromycin analogs developed for the antibacterial activity by the help of that computational approaches. So these are the list. Some list, there is more drugs are also available that develop through that computational approaches. These are the some list. So, so finally, what is the drug discovery? Drug discovery is a multidisciplinary and that is complex and costly, and some that is intellectually expensive process. But modern drug design techniques can make drug discovery process and then fruitful and rational. Means that is computational approaches. By the help of computational approaches, we can discover the more fruitful and more effective drugs. and and the knowledge of these techniques, expertise in these techniques, we can save the time as well as the save the cost of the development of the drug, because we can reduce the time, we can reduce the process of the discovery by the help of computational approaches, so we can reduce the cost of the development of the drug by the computational approaches, or we can join the different computational approaches for the development of the new drug. So finally, some softwares are also here. Just I have written these that is Tripos Compa are used for the compa analysis. Volsurf catalysts are also used. Serious and that QSR Plus is there. Schrodinger is there. Discover software is there. uh, Wheel Life Science software is there. So there are different softwares are available, and the cost of the software it is dependent on the feature of that particular software. So some use for the only 2D QSR, somebody is used for 2D plus 3D, and some software, by the help of some software, we can calculate, we can detect the compound of that. We can check the last of there is so many techniques. So by the techniques or the by the features of that particular software, there is some difference in their cost. Some software are freely available in the internet, so we can start from that part. software available, and then we can, if we have the expertise, then we can get that uh, these type of software. We can purchase these type of software. So thank you very much for listening me. If you have any query, you can ask.
Thank you so much, sir, and for sharing your experience with us. And so I want to request all the participants, if you have any query, just let me know in the chat box, or you can unmute yourself and you can ask directly. I think, sir.
Hello, sir.
Mm, Haji.
Uh, sir, when we are talking about molecular docking, you told us that it will generate a docking score.
Yes,
So I have used Autodock. It will generate like 8 models and different docking scores.
yes,
But those protein when I docked in Pymol, these proteins are very much far apart, not touching together. So how would I like
okay. It means there
understand
is
it?
yeah, difference in the results. You can say you are saying about the
Yes, sir.
difference in the result and Pymol and the dog or to dog, just like
Yes,
that.
yes,
Yes.
yes.
So we have to validate, and we have to optimize if these type of results are obtaining. Some software is saying different thing and some software is different thing, so we have to optimize by the help of different or more effective softwares. That we can use the glide software from the Schrodinger. So by the help of these, uh, we can check which compound is fitting or not, because definitely there is some variation in the results obtained. So we have to optimize and we have to validate from by the help of different more approaches or the different more effective software, we can say. So this is the we have to solve, we have to validate.
Okay, so and I have another question.
Yes,
Sir, I was trying to dock 2 proteins. So
yes.
one I selected the larger protein as macromolecule and the smaller protein as ligand. So while docking this the after docking, the autodock will prepare a protein, but it will have only small number of amino acid, not the whole protein. So how to check whether matlab, if my thing is कि I want to see where are those two are binding. So can we do anything to show to see where these 2 proteins are binding actually, or have another binding sites?
Either we have to check. We can first we have to check from the different software. I think we can check by that. I'm not working in that particular binding of the two proteins, but I think we can check by the most effective. I think that you can use the glide software. That is better one. So you can perform these study, I think.
Okay, okay, sir. Thank you.
Okay, okay.
Sir, we have another question
Yes.
from someone, GS. Good morning, sir. How is the drug design market at present? Also, what are the opportunities for drug design careers as bioinformatic graduate?
Uh, now these days, different companies is developing some. We can say that molecular modeling or the computational area. Just you can see just like the formulation development, AMD, and just like that formulation AMD, they are in the R&D department. There is one other computational uh, development area is there, in which they are using these techniques to develop the new drugs. So some companies which is working, which are working in that area of that development of the new drugs. So they are emphasizing to use the molecular modeling technique. So definitely, if you work on that drug designing, so it will help and you to get that job in that company that work in that particular field. So some companies you have to search which companies are involved in that in these area or techniques, so you can get your career in that particular um, area and in particular job in the company.
Thank you, sir. And another one is, it's not a question, but it's a query. Sir, can you give us a complete list of free softwares and licensed softwares available for molecular modeling? Okay, so we will provide the recordings to everyone. Okay, so sir already presented all the softwares and list of softwares and licensed softwares. So you can extract the data from there and okay. So and sir, there is no further question from the participants.
Okay. Thank you for
Thank you so much, sir.
everyone for listening
So
me.
I would like to welcome Dr. Karthikeyan from IGNTU. He also joined us this webinar. So I would like to invite Dr. Karthikeyan for vote of thanks and share your experience with us about this webinar. Sir, am I audible? Yes, sir.
Yeah. Good morning, all. Thank you, Mr. Monu Shukla, for providing me this opportunity.
Thank you, sir.
I would like to express my profound gratitude to the speaker, Singour sir. Hopefully, I think we will meet sometime when I come to Bhopal. And
Yes.
it was an enlightening session on drug discovery. And he had explained it so in a simple and thought-provoking manner. I hope that all the participants will be benefited from his lecture. And I am looking forward to the next lecture. So with this, and also I would like to thank the participants for asking, for being so interactive. And thanks again, Mr. Molo.
Thank you
Thank
so
you,
much.
Karthikeyan sir. We will meet definitely when you will come in Bhopal.
Sure, sir. Thank you very much.
Thank you so much, sir,
Thank
for
you.
the words, and stay tuned with us for with the same link. And I would like also welcome Dr. Harshita Tiwari for vote of thanks.
On behalf of DCRC Lab, I would like to express my sincere gratitude to Dr. Singour sir for his informative and interactive lecture. I believe that he might have instilled all the participants with the knowledge of computational drug discovery, and we will surely be benefited with the knowledge of computational drug discovery. So thank you very much, sir, for having us.
Thank you so much, ma'am. Thank you so much, Professor Dr. Pradeep Kumar Singour, for joining us and giving your precious time and sharing your experience with us. Thank you so much for experience, sharing your experience. Hope all the participants take the experience and learned something new from your slides. Thank you so much, sir. Thank you so much.
Thank you, Monu sir. Thank you.
Now, thank you so much for joining for the first lecture. Now we are moving towards another lecture from Dr. Harshita. And so I just want to give a brief introduction about Dr. Harshita Tiwari. Dr. Harshita Tiwari has a strong educational background and extensive research experience in the field of pharmaceutical sciences with a focus on computational drug discovery and molecular modeling. She worked as a senior research fellow at the Discovery Informatics Lab, Natural Product and Medicinal Chemistry Division of CSIR, Indian Institute of Integrative Medicine, located in Jammu. Dr. Harshita's dedication to her research and her contribution to various projects have earned her recognition and respect in the scientific community. Currently, Dr. Tiwari is an accomplished computational chemist at Celaris Therapeutic, Austria, and computational research scientist at Drug Chemistry Research Center. Dr. Harshita's educational journey includes a Bachelor of Pharmacy degree from Kumar University and Master's degree in Pharmacoinformatics from the National Institute of Pharmaceutical Education and Research in Kolkata. She is also on the verge of completing her PhD under the guidance of Dr. Amit Nargotra, principal scientist at CSI IIM Jammu, where her research has primarily concentrated on utilizing computational methods to identify and elucidate the mode of inhibition of potential inhibitors of E. coli, UDP, and acetylglucosamine enol pyruvyl transferase MUR-A enzyme. Her work extends to several other areas, including drug repositioning for neuroinflammation, molecular mechanisms of anti-inflammatory compounds, and database development. Dr. Harshita Tiwari's research contributions are evident through her numerous publications, and her expertise includes molecular docking, molecular dynamic simulations, pharmacophore modeling, artificial intelligence, machine learning, and data analysis. She has also actively participated in conferences, workshops, and training programs related to drug discovery and computational biology. Her dedication to advancing pharmaceutical research and her accomplishments make her a prominent figure in the field of pharmaceutical sciences and informatics. So we all welcome you, ma'am, for the today's webinar on revolutionizing drug discovery, the power of machine learning. So over to you, ma'am, and thank you so much for joining us.
Thank you, sir, for this warm welcome. Let me share my screen first.
Yeah, sure, ma'am. Now you are the host. You can present your slides.
Yeah. Can you see my slides?
Yes, ma'am.
Okay. So thank you very much, sir, for having me in this workshop, in this conference. So the topic of my today's webinar is revolutionizing drug discovery, the power of machine learning. So let me briefly introduce myself. My name is Dr. Harshita Tiwari. I am working as a computational research scientist in Drug Chemistry Research Center, dcrc lab. The flow of the webinar will be like this. So first I will introduce you with the basic artificial intelligence methods. Then I will quickly move to applications of AI at various stages of drug discovery research, majorly focusing on early phase drug discovery. Then I will conclude the important with telling you about the important AI based tools for drug discovery. Let's move toward our first session. So, by definition, artificial intelligence is a field which aims to provide intelligence to machine, which means that by the application of the artificial intelligence, we can train our machine for making predictions or for the classification related tasks. So, the field of artificial intelligence, it basically compasses of two parts. One is machine learning, which applies the statistical learning or statistical equations to achieve the task of artificial intelligence, and then the deep learning, which is a subset of machine learning techniques based on the deep neural networks for mimicking the behavior of the human brain. Then it can perform two major tasks. One is the classification task and one is the regression task. So if I'll give you an example, the classification task is the task by whose output is which gives the binary output, like it can give output in the form of 0 1, yes or no. So suppose there is a student which has appeared in an exam, and we need to have a classification task. So how would we do that? We will simply classify, we will see its marks, and we will classify either he is pass or fail. Then if you want to perform a regression task, then what we will look, we will see what is the percentage of mass. So the thing is, if we perform the classification task, the output is always binary, and when we perform the regression task, we get a it can perform the clustering task as well. Suppose we have a class of student which have different properties. Some are good in arts, some are good in music, some are good in maths, or some can be good in science. So with the application of the clustering, we can classify the students which are good in arts, which are good in maths, or which are good in science. So this is basically the grouping of the student based on particular traits. So till now we have learned that machine learning can perform three kind of tasks. One is classification, another is regression, and third is the clustering. Then it can perform these tasks by three types of learning. The first two are supervised and the unsupervised learning. So supervised learning is the learning when we have a label data. So what is the label? It means that we have simply named the output of some we have input data. Like I have shown here the few apples as an input. So here I have annotated these fruits like the apples as an apple. So I will say the model that if a fruit is red, it has a leaf and it has a little pedicle attached to it. Then it can be an apple. So I have trained the data with the labels. So this kind of learning, when we have a label data or name data, it is called the supervised learning. Then the unsupervised learning. Suppose I have given a data set of fruits to a machine learning model. So here in my data set, there are some apples, some bananas, and some peaches. So I have not tell my model which fruit is apple, which is banana, and which is peach. But I want my machine learning model to look at these fruits and classify itself the fruit as in three different categories. Then this type of learning will call the unsupervised learning. So how it will do that? It will simply look at the leaves first. So it will look at the apples, peaches, and the banana. Since banana has not leaf attached to it, so it will remove it from the category in this category. So it will group it as another subset. Okay, then it will look at the apple and the peaches. Then it will look that both have leaf, both have some kind of round shape. Then it will say that then it will move toward this task and it will look at the color of the fruit. So it will say that there are some fruit which are bright red in color. They have one category, and some fruits which appears as an orange in color. So it will classify those fruits in the different category. So this is called the unsupervised learning. When we don't tell our data, we don't give any label to our data. Then the third kind of learning is a reinforcement learning. So here I have added an example of a baby. Suppose you have a little baby in your house and he has freedom to move any part of your house. So what he will do, he can move towards the fire or any dangerous situation, or he can move towards the food. So whenever a baby goes towards the milk water or towards the food, he had some good experience. Okay, or you can say it adds some reward to the baby, and it learns that if he will go towards the right side, he will get some food. Similarly, if he chooses to go toward the fire, okay, or any dangerous situation, what if it will learn? It will learn that whenever he will move towards the fire or dangerous situation, he is harming to himself. Okay, so he will add a negative impact or negative rewards to the baby's mind, and he will not go towards that side. So this kind of learning, when a person is learning or a model is learning from its experience, or it is adding some reward to the positive situation and some negative rewards to the negative outcomes. This kind of learning is known as the reinforcement learning. Okay. So what is we in the reinforcement learning, where we provide the input data, some raw data to the model, then we add some reward Okay, so based on the rewards, we provide the output. So this is the third kind of the learning. So now we have learned that there are three kind of learning task here. One is supervised, second is unsupervised, and third is the reinforcement learning. So reinforcement learning is basically takes important role while we are dealing with some robotics. Then the deep learning. So deep learning is the part of the machine learning or the subset of machine learning which mimics the behavior of the human brain. So as we know that human brain consists of the neurons, which have some dendrites, cell body, axons, and synaptic terminals. So these elements of a human neurons help it to interact with some other neurons in the brain, because brain is a very complex structure consisted of millions of the uncountable number of the neurons. So these structures of the neurons helps neurons to interact with each other. Similarly, the artificial neural networks or the deep learning methods consisted of several input layers, which we call the neurons, which interacts with each other and provide the output. So this kind of structure is known as the artificial neural network. So it is able to learn without human supervision, drawing from the data that both unstructured and the unlabeled data. Then the soul of the machine learning model or the artificial intelligence. So the basically the all the machine learning models are depends on the data. So if we will provide a good quality data to our model while training, it will resulted in a good quality of the model. So whenever we provide a data to our model, it should be accurate, complete. It should be in a uniform format, consistent. It shouldn't have any duplicate, and it should be integrated. So if you will provide a good data to your model, then it will result in an accurate results. Then there are basically 7 steps of the machine learning. First is the gathering of the data. Second is the preparation of the data, in which we clean our data by removing the duplicates or fill the missing values. Then once we have a good quality of the data, then we choose a machine learning algorithms to train our model. Once we have trained our model with the provided data, then we evaluate our model with respect to the accuracy and its predictability. And if we don't get the accurate results, then what we do, we perform the hyperparameter tuning. So hyperparameter tuning is nothing but it is the set of some parameters on which the performance of the model depends. So we just uh, alter the parameters of our model, and then check the predictability of our model. Okay, once we got some accurate model, then we use that model for the prediction. So there are various types of the machine learning models, and which has application in different fields. So we can use it's for the diagnosis purpose, for image classification, for recommendation system, or for a game skill acquisition. So different kind of learning has different application, and it has got the wide range of application in today's context. So this was about a basic introduction of machine learning techniques in general. Now I'm moving towards the application of AI at various stages of drug discovery research, and I will majorly focus on the early phase drug discovery. Now, let me give you a brief introduction of drug discovery process. So it is basically it can be divided basically in the 3 steps. One is the drug discovery, which is early phase drug discovery, then the clinical testing, and then the approval for the production. So the first phase of the drug discovery, it is basically consisted of target identification, lead discovery, lead optimization, and the preclinical testing. Then the second phase of drug discovery is the clinical testing in the human beings. Then once the drug we have the results from the clinical testing, we provide the results for the approval to FDA or EMA. And once the drug is approved or in market, we perform some surveillance on the drug's performance. We check if the drug is having some side effects or not, or if it is performing any good, it is providing good results to it. So this is the overall process of the drug discovery, and it is a very long and time consuming process, and it is very costly too. So whenever we are performing the computational drug discovery or artificial intelligence drug discovery, our aim is to reduce the cost as well as the time in the process of the drug discovery. Then this process of the computational drug discovery can be divided into basically two parts. One is the bioinformatics and another is the cheminformatics. So bioinformatics is the process by which we are gathering the data from the biological targets, so which can be proteins, DNA, RNA. Okay. Then the cheminformatics is the process in which we are getting the information from the large chemical space or the compounds. So there are different compound repositories which are freely available as well as purchasable, which help us to perform this cheminformatics task. So for a successful drug discovery processes, we have to take both the processes together. Then, in context of the drug discovery, AI refers to the utilization of the computational algorithms and machine learning techniques to analyze the vast amount of data, identify patterns, and make predictions. Okay, so from where this large amount of data comes from to build the machine learning models for the drug discovery. So, this drug discovery. So, before moving towards this, I would like to quickly um, take you towards the different task of the machine learning I have told you, with context to the with context to the drug discovery. So I have told you about the classification task with respect to the classification of foods, and now I want to now in context to the drug discovery, suppose I want to classify the a set of kinase inhibitors. So if I need to classify this model, and I have a data set of compounds consisting of kinase activators and the kinase inhibitors. So what I will do, I'll train my model. It can be supervised or unsupervised with this data set, and I will simply, after training, I will provide the unknown data set, and it will tell uh, if whether these compound is a kinase inhibitor or not. Okay, so this is the classification task. Then the regression task, as I have told you, the regression task is the task by which the output is a continuous number. Okay, so here if in context to the drug discovery, how if I want to perform the regression task, what I'll do, I will provide the data set of compound with the set of some values. Okay, here I have provided the log PW values or log P values of the compounds, like 1.77, 2.1, 2.2, 1.64. Okay, when I am providing the compounds with these values, and now I am training my model, and I am building a model and use it for the prediction of certain log P value. Okay, then it comes under the regression task, by which we are deriving the value of log P. Okay, and this is the regression task. Now, if I want to perform the clustering or the clustering task with the respect of this, what I'll do, I'll simply provide the set of kinase inhibitor, and I don't have labeled these compounds with any activity. Okay, so I don't know whether this is a kinase inhibitor or not. Okay, then what the model will do, it will simply look at the scaffold of these compounds, and then it will cluster the compounds based on their scaffold. So this is the classification example with the unsupervised learning. Then the application of reinforcement learning in the field of the drug discovery. Okay, so suppose I have some compound. Okay, some scaffolds, like this is the cyclohexene ring or cyclohexene ring, with some methyl group or the oxygen ring attached, atom attached to it. Okay, so here the agent is the scaffold. So I have this scaffolds of the compounds. Now what I can do, I can either add some scaffold to the compound, remove or replace the atom. Okay, so each time whenever I will add, remove or replace the atom, it will have some effect on the activity of the compounds. Okay, so here if it will increase the affinity, so the reward here is if the affinity is increased, then I'll say this addition is the good. Okay, or if any addition decreases the affinity, when what I'll say it is not good for the new molecule. Okay, so what I will do, I'll simply add, remove or replace some atoms in the molecules, and then I'll check which function is increasing the property. So based on this, I will just derive or generate a new molecule for a certain particular targets. Okay, so this task in which I am using a particular reward or the penalty function, this is the example of reinforcement learning in the field of drug discovery. I hope you might have understand this concept. Now, as I have told you, there are 7 steps of the machine learning. So, in context of the drug discovery, the first step is the gathering and preparation of the data. So, in the drug discovery, the data comes from various experimental as an outcome of the various experiments. So it can come from the scientific literature, which scientist has reported from their previous experiments. It can be the patents related data. It can be the data related to the clinical trials in which a compound has failed, or it has got approval for a particular kind of activity. It can come from the marketed drug. Either the kind of drug has performed well or it has created the side effects. Then the small molecule crystallographic data, like the data might have come from the crystallographic experiments or the or the experimentation techniques. Then the protein ligand complexes related data. So this kind of data might have come from the docking experiments, from the crystallographic experiments, from different which is stored in the different databases like protein data banks. It can come from the scaffold or the isostereos. Like we have performed a particular pharmacophore based assessment of the compounds or the QSAR based assessment of the compounds. We have performed a bioisosteric replacement of the groups in particular compounds. Okay. From this, the different kind of data we gather to build the models for the drug discovery. Okay. So now the data is in raw form. Our task is to remove or to prepare the data for the building the models. Then once we have set of compounds from these data sets, we have to calculate the molecular descriptor of the compounds. So in general language, these are some properties of the compounds which describe the compounds. So molecular descriptors can be 1D, 2D, 3D. So in terms of the 1D descriptor, it just tells about the elemental composition of the molecule. For the 2D descriptors, it tells about the number of hydrogen bonds, number of hydrogen bond acceptor, log P values of the compounds. And in terms of the 3D descriptors, it tells about the properties which basically depends on the 3D structure of the compounds. So they can be molar surface area, molecular volume, etc. So these are the molecular descriptors we need to calculate from the prepared data set, from the collected data set of the compounds. Then once we have these molecular descriptors, like here's an example of development of ML, a model of solubility prediction of the compounds. I have the training set of the compounds which have some solubility value. So I need to calculate the solubility of the compounds of the compounds. So these features are known as the molecular descriptors. So I have calculated the molecular descriptors, that is molecular weight, hydrogen bond acceptor, hydrogen bond donor, and the ring feature of these compounds. Okay, so as you can see that Molecule number 1, the molecular weight is 300. For molecule number 2, the molecular weight is 450, and so on. Okay, so you can see that the solubility profile of these molecules, it is also changing. Okay, as the molecular weight is changing. Okay, similarly, the solubility value of the compounds or the molecules are also changing as the hydrogen bonds are changing. Okay, same in the case of the hydrogen bond tuner. But when you see in case of the ring characters, okay, either the solubility value is 1.23 or 1.55 or 2.0, 0.3. The ring number of ring is not changing. So we can say that the property means the property which we need to predict like solubility, it is not depending on this ring feature. Okay, so we can remove this feature to minimize the errors while developing the model. So this process of removing this unwanted feature while building the model is known as the data cleaning or data processing. Okay, now we will build the model of solubility prediction with the help of the three features which are important for the solubility predictions. Now, what we have done till now, we have collected the data set of compounds, then we have calculated some descriptors, and we have removed the descriptors or the feature on which the solubility value is not changing or not depending. Okay, now we can build the model, then evaluate the model. Okay, now you can see that I have divided the data set for the model building in two sets, like one is the training set and another is the test set. Okay, so training set is the data set of the compounds which we used for the building of the model, training of the model. Once we have built the model, we can use the test set for evaluation of that model. So this is the overall example of development of a model with the help of machine learning. Then the application part of the machine learning. Okay, so it has a wide application from target identification and validation, from compound screening and lead discovery, from preclinical development to clinical development. Okay, so for each task we need different kind of data. Then this is a landscape of the companies which are working or using these drug discovery of machine learning or artificial intelligence technology for different tasks. So the first one are the companies which are using these techniques for early phase drug development. The second phase companies are using the AI based models for the preclinical development, and the third group of companies, they are using the AI based models for clinical development. In addition to that, some of the companies, and they are the very big pharmaceutical players, they are using the AI based techniques for end to end drug discovery development. Means they are using the AI based drug tools for all these three processes. Now, some of the breakthroughs in the field of the AI based drug discovery. So there is a company which is known as the AI in silico medicine. So they have used their generative AI. So generative means they are using they are using reinforcement learning for the generation of the molecules. Okay, so in 46 days they have generated and a compound with the help of these AI techniques in 46 days, and they have checked those compound in vivo models as well, and the compound was active for the cancer. Then another breakthrough is DeepMind's AlphaFold. As we know that protein folding is a very important task, because the structure of the protein depends on the structure of the function of the protein depends on the overall structure of the protein. Okay, so the DeepMind is a company which is using these artificial intelligence or the deep learning tools for the drug discovery purposes. They have designed a tool AlphaFold, which can predict the the structure of the protein with 90% accuracy. Then another breakthrough is the first breakthrough was the by the Extensia. So it is the company which is using the end-to-end AI techniques for the drug discovery. So they have announced their first ever AI designed drug molecule to clinical trial in early 2020. Then in July 2021, the DeepMind announces the AlphaFold model to predict the 3D structure of the protein. Then in February 2022, in Silico Medicine, they started the clinical trial for the AI based developed drug, and in February 2030. So this drug, which in silico medicine has brought to the clinical trial, it got the orphan drug status. Okay. Then another breakthrough is in January 2023, AB Science, they have designed an de novo antibody. So de novo means from the scratch. Okay, so they have developed the de novo antibody and validated it in validated is. So they are using generative AI for this purposes. Okay, then this is the breakthrough for the related to the AlphaFold. Then the recent example or the recent breakthrough is the alpha missense, which is again proposed by Google's DeepMind. So what they have done. They have they have created a model which can predict the missense or the mutation which can lead to the pathogenicity of which can lead to the pathogenicity. So what will be the future perspective of utility of this model? Suppose as we all have suffered from the COVID pandemic, okay, in future we also encounter some new pathogens, and we know and nothing about these pathogens. Then and we have such tools like alpha missense, so we can check the genome of this new pathogens, and we can predict which pathogens means which kind of mutation it can have in the future, and and which mutations can be pathogenic. So we can design the antibiotics or the vaccines in advance to avoid any fatalities in future. So these are the some breakthroughs of the AI in the drug discovery. Now moving towards some important AI based tools for drug discovery. So now, as we know that the process of the drug discovery starts with the with the literature survey. So, and the literature survey, it is a very cumbersome task, and it should be very accurate, and it is it is impossible to go through all the papers and papers for designing your study. Okay, so there is a tool which is known as the Elicit. So it is an AI based tool which can but if you will give like I have provided the keyword EGFR here, it as soon as you hit the search button, it will give you all the papers which are related to EGFR, and it will provide the abstract summary of the paper, and it will also provide the interventions like what drugs are used in this paper, what are the outcome measures, like what is the IC50 of the compounds, what is the MIC50 of the compounds, okay, how many number of participants in this paper. So this tool is very useful and designed with the AI, which you can use for design your study and for the literature survey. Then another tool which you can use is the AlphaFold tool. So as you know that our body is made up of proteins as a macromolecule, and these proteins serve in different functions. So these proteins represent a very good target for designing any novel drugs. Okay, and for designing or targeting any drug or the target, we need to have some the information related to its structure, because the structure depends on the protein. So the structures can be divided into primary sequence, then primary sequence, structure can be divided into primary sequence, which is the arrangement of the amino acids in a string. Then the secondary structure, when these amino acids interact with each other by the help of hydrogen bonding, it gave rise to the secondary structure. Then the tertiary structure, which is the arrangement of the secondary structure in the 3D space. Then the quaternary structure, which is the arrangement of these tertiary structure for a functional form, like in the case of the hemoglobin. Okay, so this information is very important for targeting a molecule. So this it is it is easier to get the information related to this primary structure. But when comes to the tertiary and the quaternary structure, it is very difficult for some protein to derive this structure. Okay, so there are some experimental procedures by which we can derive the structure of the proteins. Some is X-ray diffraction, NMR, and the cryo-electron microscopy. But the major disadvantage of using this model or this method, experimental techniques, is that these are very difficult, which needs expertise, as well as it is very time consuming. Okay, so we cannot quickly give any protein sequence or protein to these methods and say and derive the structure quickly. Okay, in that case, like I have discussed earlier, that the structure is function depends on the sequence. We can use the some predictive softwares like iTester modeler, Swiss model, and now the AlphaFold tool, which is an AI based tool for the to get the information related to the protein structure. So the accuracy of this structure is 97%, and they have created a separate database of the structure which are derived from this AlphaFold tool. So if you are not getting any information related to target in protein data bank, you can query the structure in this AlphaFold database, and you can get the information related to the 3D structure of that protein. Okay, so this is very important tool for the drug discovery. Then the third is the Vega. So, as you know that for the development of these machine learning or AI based models, we need to have some expertise in the programming, okay, or expertise in the programming languages for the implementation of these tools. So, in that case, when a person doesn't have any experience or prior knowledge to the programming languages, they can use Vika. So this is basically a GUI based tool by University of Ricardo. So here you can provide your data. It can perform all the tasks related to machine learning, like data processing, pre-processing, cleaning, visualization, and simple machine learning model building. So these are the important tools for machine learning model building. Then if you have knowledge to the programming languages, then you can use a specialized library for machine learning model building. So the first one is the RDKit library, which is very important for cheminformatics related tasks. Here you can calculate the descriptors, you can perform the basic mathematical operations, you can visualize or convert the molecules to another library is the deep chem library. So this is basically for the cheminformatic purposes. You can visualize your molecule, you can convert the molecules in different format, or you can also minimize your molecule with the help of deep chem library. Now we have these library, and you want to implement these library, you can use the Anaconda software, which comprised of different programming platforms like R, Jupyter, for the implementation of or the building of your machine learning models. Okay, so these are some of the important tools which you can use for your research. Coming towards the conclusions. So as you know that these are there is an increasing demand of these tools in the field of drug discovery, and they have provided a very promising result to augment the overall drug discovery processes. As you can see that in silico medicines have generated and evaluated their molecule in 46 days. Then with the accessible framework, now researchers there is no they have overly developed the tools for the drug discovery. Researchers needs to get involved more in testing validated so that robust tools can be developed. So there are different tools now available by which we can implement these methods. Okay. With this, I would like to thank all the participants, and I hope this sessions will be informative for all of you. Thank you.
Thank you so much, ma'am, for sharing your experience with us. And I welcome all the participants to share their queries in the chat box, or you can directly ask by unmuting yourself. Machine learning is a very trending field of nowadays for basically for drug repurposing and drug discovery. So if you have any query, just let us know. There one query. It's a not a question. Ma'am, could you give in hand on training on those softwares? So definitely we are organizing time to time training programs. So hopefully in the coming week or month, we will organize a training session. Okay, in the machine learning based softwares or different softwares for drug discovery. So stay tuned with us, Mr. Gaurav. Okay, and any query from different participants who joined this webinar? Ma'am, I think you taught very well, so there are no queries.
Yeah, hello.
Yeah. Dr. Karthikeyan has a query. Yeah, yes, sir. Go ahead.
Yeah, thanks, Mr. Manu. Thank you, Dr. Harshita. It was a very, very informative session. I have one query. Can you elaborate more on the application of machine learning in target prediction in drug discovery
Yes, sir.
using
So
the
actually,
using the genomics data?
yes, sir. So now, as we know that genomics is it is a very old means it is very well studied subject. So we have lots of data from human genome sequencing, as well as the technologies are advancing, we are getting more data from the genomics experiments. So we can analyze these data with respect to the expression of certain genes um, in particular phenotypic situation, like in particular disease situation or healthy situation, by which we can predict which genes are important for a disease progression, and which doesn't play any role in the disease progression. And we can also go for the precision based or the gene based drug discovery with the help of these machine learning data. So as I have mentioned extensia, so they are using these genomics based studies in their lab, and they are applying these methodologies for precision based medicine designs. So they are working very well in this field. This is the application part
Yeah.
of.
So
Yes.
one interesting area I would like to explore is the genes responsible for the development of resistance. And since you have good experience in antimicrobial drug discovery, for example, treatment of different organisms or different bacteria with antibiotics leads to the development of resistance. So, do you have? Is there any database which is available, which gives you information about the expression of genes after the treatment of antibiotics? Are those genes responsible for the development of resistance?
Sir, I don't know. I don't have any information related to the name of these, but the Google Missense tool I just mentioned in my presentation. It might be helpful for this purpose, because they have predicted this. They have used this tool to predict the pathogenic mutations or the treatment which leads to pathogenic and which doesn't have any role in the disease progression. So this tool can also be used for the studying the disease response or the resistance of the drugs in particular microbial infections too.
Okay, doctor. Thank you very much.
Thank you, sir.
Thank you so much, sir, for putting your query with us. And ma'am, we have a query from Mr. Sagar Rajput. Drug discovery had become more effective due to AI, but still it take lots of time in preclinical trials and clinical trials. Does AI can be also used in preclinical and clinical trials to make it more time effective?
Uh, so with the help of these AI techniques, we are minimizing time in right. In future, I believe that there will be some models which can make the behavior of animals too. I'm not sure about the humans, but for the animal behavior they can mimic. So we can reduce the time in the preclinical development of the drugs with the help of these AI based tools.
Okay, ma'am, I think you are also presented in your slides that there are various companies who are working for clinical trials and preclinical. Okay, uh, in the last slides.
Yes,
So,
they are
so
working.
this is yeah, this is possible. I think already possible. Already many companies are working on it. So
Okay.
we have another query from Mr. Gaurav. Ma'am, I have a doubt that what strategies are being developed to overcome the data security problem in drug discovery, analysis of a drug with machine learning, especially in rare disease.
Okay, so that was not data security. I guess this is data scarcity. Overcome the data scarcity
Scarcity, yeah.
problem in drug discovery analysis of drug with machine learning, especially in rare disease. So I don't think there is a data scarcity because lots of research has been done. So now the thing is that we need to develop some uh, accurate models and we need to work on the data cleaning. Okay, one thing is that. Second thing is that we have some positive data because most of the researchers they report the positive data, but we don't have negative data. Okay, so data imbalance is another aspect. So if we will start reporting our negative result as well, then the problem of the data imbalance that that can be solved. Then moving towards the problem of applying these methods in the rare diseases. So, if you look from the perspective of the companies, okay, so companies are really working in the means they really rarely work in the field of rare diseases because they don't get much profit from that. Okay, but I was talking to some of the people who are working in this area. So now people are means they are increasing the funding in the field of rare diseases. Definitely, the field of rare diseases based drug discovery will also bloom. So yeah, that was my answer for your question. I hope I have
Thank you
answered
so
your
much,
query.
ma'am. And is there any other queries from the participants? Okay, there are various renowned personalities have been joined our webinar, like Dr. P.K. Singour, sir. If you have any query or you want to share your experience with us, sir.
Uh, no, I'm just listening because I'm also learning about the machine learning. So Madam told very good manner and very good way. I'm just ask one thing. I have one query that can there is some freely available software that regarding the artificial intelligence in the drug discovery. There is some freely available software or not?
Sir, most of the tools are free for the
Okay.
artificial intelligence. So if you have some knowledge related to programming, then you can go for the Python or Anaconda, and you can use the libraries like DeepCam and the RDKit for the implementation or building of the model. And if you don't have, you can use Vika. And for the and there are some tools for the docking studies as well, like Deep Dock. You can use for that, for machine learning based docking studies as well. Yes,
Okay. Thank you. Thank
yes.
you, madam.
Thank you, sir. Sir, I think we cannot hear you, sir.
So, ma'am, there is no query from the participants. Okay, so now we are going to bind up the webinar, and I would like to welcome Dr. PK Singour, sir, for vote of thanks.
I would like to thank Dr. Harshita Tiwari, ma'am, for explaining well, because I'm also working on drug designing, but I'm new in that machine learning and artificial intelligence. So I'm learning. I was listening very carefully, so I can then implement, and I will learn also from you when you will organize that hands-on training. So I think all the participants will get the knowledge, and then they will utilize the concept in future also. So I thank you very much, ma'am, for that giving the times to all to sharing this knowledge. And I thank to Mr. Monu Sukla sir for inviting me also. So thank you all very
Sir, I'm still your student. Don't call me sir.
much. No.
Thank you
Okay,
so much,
all is
sir.
sir. Okay, thank you.
Thank you so much, sir, for sharing your experience with us. So I would like to welcome Dr. Karthikeyan to give a vote of thanks and share your experience with us, sir.
Thanks, Mr. Monu. It was a great opportunity for me to listen to Singour sir and Dr. Harshita. And it was a very relevant topic and very up to date. So it's quite interesting to see so many students attending this seminar, because drug discovery, as you know, it's a evolving discipline. So it is always good to be up to date with the recent developments in this area. And machine learning is a very interesting area, and in which I think our country has a lot of potential. So training the students in this very important area would be a great contribution to our country. Thank you once again, Monu and Dr. Arshita and Singour sir. Thank you.
Thank you so much,
Thank you.
sir. So it was not a national webinar; it was an international webinar. One of the participant from US, Mr. Subham Pandey, was here in the webinar. So I would like to welcome Mr. Subham Pandey to share your experience with us. Mr. Subham, are we audible for you? He is from Alabama University, from US. Okay, so there's a query from Subham Pandey. Thank you, ma'am. If possible, please arrange some workshop on training in machine learning. So I just want to tell every participant of this webinar that we are going to organize some training programs on CAD, computer-aided drug design, as well as machine learning for the drug discovery. Okay, so stay tuned with us. We are timely organizing webinars, training programs, and conferences. So you can time to time check our website, that is dcrc.co.in. Okay, so and we are also welcome to all the participants. You can contact us for collaborative work, as well as training sessions. So we will also organize some training session and webinars with your institutions and universities. Thank you so much, ma'am, and all the participants to share your experience. Okay, so ma'am, I would like to share one slide which is related to the certificate of appreciation. So uh, please make me host so that I will be able to present my slide. Okay, thank you
I
so
did
much,
mute
ma'am.
you.
Thank you so much, Dr. Harshita Tiwari. She is also working as a computational research scientist at DCRC Labs, as well as she is computational chemist at Celaris Therapeutics. Thank you so much for sharing your experience with us. Hopefully, this will webinar will be fruitful for everyone, and it's a trending topic for current drug discovery. So thank you so much, ma'am, and all the participants for joining the webinar. Thank you so much. The certificates and recording will be provided you within a week. Thank you so much.
Thank you.